Hi, in this video I am going to speak about some of the basic commands which are used in bioinformatics when you deal with large sequence files, especially in the formats like fasta and fastq files. So it's very useful to know the Linux command, so that when you do the analysis or when you are trying to run when you are planning very big experiments, these kinds of simple commands in the Windows might not be helpful to you. So using the Ubuntu or Linux flavor, using the Unix commands, it would make the life very much easier when you have very big data sets. So especially when you deal with the sequence files such as fasta files and fastq files. So in this video I am going to speak just a little introduction about the commands which can be used on the fasta files, and once again these commands are very useful for the beginners. These are not exclusively for the very experienced kind of people. They might feel these things very, nothing, but these would be very helpful to the beginners. So let's get started. So what are these fasta files? If you are a beginner, then you might not be very much conversant about what are fasta, what are fastq files, but the fasta files are those files where the nucleotide sequences or peptide sequences of certain genes are placed in a format. For example, the fasta format looks like this, of a protein sequence looks like this. So this is the symbol which represents that this is a fasta header, and this particular sequence is downloaded from the revseq. So this is the revseq, and this is the gene ID, and this is the accession number, and the description of the specific protein, and from which organism it has been found. So the next line would contain the actual peptide sequence. So this is how the protein sequences represented in a fasta format. For the nucleotide sequence, you would find instead of amino acids, you would find the sequence made with A, G, T and C. So and the next thing is like okay, so now someone asks you to download a specific gene of interest, and you don't know from where to download it. So the first thing, there are many different sources where you can get the sequence, but the popular very large collection of the sequences are found especially in two different databases. One is called revseq, and which is available from the NCBI website, and another one is Ensemble. So I would just show you how you can download or you can download a specific gene from the revseq database. For example, just go to Google, type revseq, immediately get redirected to the revseq. Now here you can download the nucleotide sequence as well as the peptide sequences also. Let us suppose I want to download a gene called SOX. So it would then list me all the SOX gene in different organisms. I haven't specified which organism it is, so it would give me all the list of sequences in the header if it contains the SOX. If let us suppose I just want from the Homo sapiens, just click the Homo sapiens, and there it goes, it connects, and then I also need only the mRNA sequence only. Instead, I mean, it sometimes gives you the genomic DNA, all those different sequences also, but suppose you want the gene which is coming from mRNA sec, then just click on the mRNA. So if you click that, it immediately goes here. So see, you contain the SOX homolog, all those things. Now okay, now if I want to download this only one FASTA sequence, then I can just go here and open the record here and click on the FASTA sequence. So see, this is stated as mRNA here, so obviously you will have the sequence which contains only AGTC. So you can just copy this and you can save in your file, or I think they have an option here, so you can click file and FASTA. Yes, so you create a file and okay, then it asks you for download, so you click save. I save it in here. Okay, this is the gene name and I replace it with SOX. So once I downloaded the SOX, I have a FASTA file, but what if I have to so this one, you see, this is a nucleotide sequence because we selected all the mRNA sequences. So what if I have to download all these 143 sequences? Same thing what we have done just previously. Go to send to file, and then click on the format FASTA and then create file. So it would give up a pop-up here, and you save as all SOX genes in Homo sapiens. Okay, good. But what if I want the protein sequences instead of the nucleotide sequences? So this time what I will do, I'll go here, I'll search the same SOX genes in the Homo sapiens, but this time in the protein here. So I search here, so I would go to this page. So now I found to have 143, but this time you see the difference. Previously all of the sequences were the nucleotide sequences here instead of protein. Now because we selected protein, all the sequences, you see the protein here. So similarly, if I want to download this, go to file and then FASTA and then create. So again it pops the download. So instead of this, protein. Okay, actually this must be mRNA. Oops. Okay, so okay, so now we have the FASTA files downloaded. Now we see how actually we can go through the Linux commands and how they are helpful in dealing with FASTA files. So if you have Linux terminal, then you can just quickly go to terminal, and then that pops the screen. That pops the terminal, and that pops out the terminal, and I just go to the directory where I have downloaded the files. So I have downloaded them on now. Okay, the first thing, okay, let me just tell you, sometimes most time okay, now when I downloaded from the RefSeq database, I did not, I mean that the sequences were not zipped, but sometimes you might get from some different sources where they zip the file because they have the size constraint. So they give you the FASTA files in this format, like they zip it. So sometimes one thing to mention is sometimes the fasta has an extension in the file name as .fasta, and sometimes it has only fa, and sometimes they also zip this, and they put the file name .fasta.gz. So anyway, the convention might be little different, but still they represent the basic format that is called fasta file. So now let us suppose I have data from this file. This is also fasta sequence file, but it has been zipped. But this is a fasta sequence file which has not been zipped. So now if you get a sequence like this, I mean the file like this, and you don't have to get panic. Then how do you deal with the data? Then if I try to open it, okay, if I try to open it, how do I need to open it then? So the command for that is, so here it states it's gz, right? So you have to gunzip upstream.fa, so it will immediately unzip it. There, now we see, so it has been unzipped. Now you don't see that .gz. Okay, that is first command. That is our first command. Okay, now that it has been uncompressed, now let us see how the fasta sequences look like. We go with the fasta file that we have already downloaded. Okay, to view the file, I want to see the contents in the file. So there is a command called cat. So if I do that, so it will immediately display me all the sequences in the file. But when if it is a small file, then it is okay. Just you can show, I mean it can show me a screen output as much as you can see through your eyes here. But I mean if the file is very large, it just rolls down to the end. So how would it be helpful to me to use the command called cat? So usually there is also another command. It's called less. So when you do less, it shows you only the screen full of output. That means when I do less the file name, it shows me only this much. This is the beginning of the file. So if I press enter, it shows me like the down region of the sequence. So as long as I press the enter, it just goes down, and it shows us the complete file. So this is the command called less. So okay, what if I just want to, so I don't want to, I just want to print only 10 lines of the file onto the screen. Then how do I do that? So for that I use a command called head -10 shows me the first 10 lines of the file, and I use the file name called also mRNA fasta, and it shows me 10 lines. So these are the 10 lines which the file. Similarly, if I want to look the bottom 10 lines of the file, I use the tail. So 10 lines of the file also mRNA in humans, again this. So I want to clear the screen, you can type the control L on from your keyboard, and or you can type also clear, both are same. So that is one command. And now okay, what if I want to edit a file? So while editing a file, if you are using a flavor such as Ubuntu or other operating systems like Fedora or something, you can always use a GUI editor. So for Ubuntu there is a editor called gedit. So to open it, I type gedit mRNA and you can open it. So it opens the file there. See, that is GUI based. What if I want to open in the terminal itself? So I can use VI editor to open it, and this shows me in the terminal. And I mean, you can also edit it by going into the insert mode. Oops. And you can exit from the open the file which you have just opened. So now once you now understood how the files can be viewed, so we just go on to the next set of commands. That is okay. Now you have been given sequences, so you just want to find I mean, you entered into a folder and you want to find what are all the FASTA files that I have now. So I type ls. Ls means list, star means all the files, dot fa sta. So I want to find out what are all the file names which end up with .fasta. So when I press that, I see these files. I have 1, 2, 3 and 4 files which end up with a name called .fasta at the end. So and I also want to find what are the sizes, and so for that I type the list and minus lh star dot fasta. So this shows you the file names and the permissions and what are their sizes and when they were modified, at what time, and the file names. And so yeah, and okay, now let us go. Okay, now I want to find or search a pattern in a FASTA file. Let us suppose I have a file, a FASTA file like this. So the FASTA file contains all the headers which starts with greater than symbol here, and a sequence from the next lines. I want to find what are all the header lines in this file. So how do I search that? There is a very powerful command called grep in the Unix. You can just type what pattern you want to search. So get grep means get regular expression. So it will now this command called grep will search for the greater than symbol in the FASTA file, and it would print you all the sequences which have all the header lines from the FASTA file. So the grep is very good. So there are some other utilities of the grep also. Now I want to find how many of sequences are there, because each sequence is supported by a header line like this. So if you count the number of headers, then it is equal to the number of sequences in the FASTA file. So you can count the number of greater than symbols in this FASTA file, and it would give you 143. As you see, we have downloaded 143 sequences here. So now similarly, if you want to find out what are all the number of sequences in all the FASTA files, and you can do like this. So it will find in all the sequences. I think there is one big file in that, so it will take some time because it is getting regular expression. So it takes some time when you use the grep. So you see that there are actually 4 files. I just showed you all these so there are 4 different files. Because these were small files, it quickly displayed the 143 here, but the third file, which is the large file, very large file, it's just around 3.5 GB. So it's taking so much of time to count the number of greater than symbols. So it shows that the grep is not very useful when you are dealing with very large files, especially now in the next video you will see you have the fastq files. So you find what you find the pattern in the if you find how many reads or the sequences are there in the fastq file, it's very difficult for us to find out how many reads are there actually in the file. I mean sequences are there in the file. If you use grep, it takes long time, so you have to use some other utilities other than grep. So for now, to just give a simple idea, I would just tell you the how you can count quickly using another flavor of grep. It's called fgrep, which is fast grep. So this one does not check for the expressions, but it just checks for the grep. So it will not it's very fast. It's faster than grep. You can see if I run the same command fgrep, it takes much less time than what the grep was taking. So we just wait. So in a way, I mean, because the files are very big, it takes much time here, but when you deal with very large files, there is comparatively very much difference between the grep and fgrep. So that is one more command. For now I would stop it because that's a very large file, 3.5 GB. So that is about the fgrep and egrep. Okay, having talked about the fgrep, I would also speak about the egrep. So egrep is something like this. Okay, I have okay, let us open a fasta file and okay, okay, I want to extract two sequences instead of one. I want to extract two sequences. So extended grep would help. Egrep is extended grep. It would let you know. It would let you to extract multiple patterns at one time. So these are the two different IDs from two different sequences, and you see how the grep will capture both of them and print it onto the screen. See, so there are two different sequences, and it displays you two different headers. So these are some of the basic commands which I usually frequently use on the fasta files. And if you want to count how many lines are there in the fasta file, then you can use word count -l and dot star dot fasta. So it would display you all the number of lines in each file, and Yeah, yeah, that's all for now. We say, if I get I mean, if I see more utilities which are helpful, then I would make another tutorial, and would upload for you. So that's all for now. Thank you very much.